We can classify neurotransmitters as excitatory or inhibitory, and direct or indirect. An excitatory neurotransmitter is one that is trying to produce an action potential in the postsynaptic neuron. So that means that neurotransmitter is trying to bring that postsynaptic neuron closer to threshold of -55. In other words, that neurotransmitter is causing that postsynaptic neuron to be depolarized. So excitatory neurotransmitters are trying to bring that postsynaptic neuron closer to threshold, and therefore it's trying to elicit an action potential in that postsynaptic neuron. An inhibitory neurotransmitter is trying to prevent an action potential in that postsynaptic neuron. So an inhibitory neurotransmitter is going to cause that postsynaptic neuron to move away from threshold, or in other words, it's going to cause that cell to be hyperpolarized. So it's going to become more negative. So neurotransmitters can be functionally classified as excitatory or inhibitory, depending on if they're trying to generate an action potential in the postsynaptic neuron, or if they're trying to prevent an action potential in that postsynaptic neuron. We can also functionally classify neurotransmitters as direct or indirect. So this is the mechanism by which these neurotransmitters are actually producing that graded potential. So direct versus indirect is just trying to explain the mechanism of action by which that neurotransmitter produces that graded potential. A direct neurotransmitter is going to bind directly to a chemically gated ion channel, causing that ion channel to open, allowing ions to flow in and out, and therefore causing a change in the membrane potential, or in other words, producing a graded potential. So a direct neurotransmitter is going to bind to a chemically gated ion channel, causing that channel to open, allowing ions to flow in and out, therefore producing a graded potential on the postsynaptic neuron. Direct neurotransmitters have a really rapid response, so they're going to very rapidly produce that graded potential because there's really only one step required in this process in order for that ion channel to be opened. The neurotransmitter just has to bind, causing that channel to open. Indirect neurotransmitters are going to act through a secondary messenger. So in this case, it's not the neurotransmitter itself that's binding to chemically gated ion channels. Instead, that neurotransmitter is going to produce a secondary messenger. That secondary messenger is then going to bind to those ion channels, causing them to open, allowing for ions to flow in and out, producing that graded potential. So indirect neurotransmitters are going to act through a secondary messenger. So in other words, that neurotransmitter is going to produce a secondary messenger, and then that secondary messenger is going to actually bind to that chemically gated ion channel, causing it to open, allowing ions to flow in and out, and therefore changing or producing a graded potential. This is going to be a slower process because there are more steps involved. It's also going to have a much longer lasting effect because that secondary messenger is going to be present for a longer period of time. Direct neurotransmitters are going to bind to channel-linked receptors. So direct neurotransmitters are going to bind directly to these chemically gated ion channels. Or in other words, their receptors are channels. So when that neurotransmitter binds, it causes that channel to open, allowing for ions to flow into the cell or out of the cell. Channel-linked receptors can be excitatory or inhibitory. So in other words, this channel might be allowing positively charged ions in which case it might be causing depolarization. This channel could also be inhibitory if it was allowing negatively charged ions in which case it's going to make that cell more negative or hyperpolarized, and therefore inhibit the generation of an action potential. So channel-linked receptors are going to open when the neurotransmitter binds, allowing ions to flow in or out of the cell, depending on if positive ions are entering or leaving, positive or negatively charged ions are entering or leaving those channels. They would be considered excitatory or inhibitory if they're making that postsynaptic neuron more positive or depolarized, or more negative or hyperpolarized. These receptors are known as channel-linked receptors. Some textbooks will also refer to them as sonotropic receptors as well. So channel-linked receptors are going to be very direct, right? So we're going to have the direct neurotransmitter binding directly to that channel or to that receptor. Because this is just one step, it's going to be a really rapid process. So it's not going to take very long at all for that channel to open once that neurotransmitter has bound. This is going to produce a very brief graded potential because the neurotransmitters are going to be very quickly degraded, diffuse away, or they're actually going to be recycled back into that presynaptic neuron. So channel-linked receptors are for direct neurotransmitters, produces a very rapid response in the postsynaptic neuron, or in other words, there's a very rapid graded potential produced. But this graded potential is extremely brief because the neurotransmitter is only going to be present in the synaptic cleft for a short period of time. Again, neurotransmitters in the synaptic cleft are going to diffuse away. They could be enzymatically degraded, or they could be recycled back into that presynaptic neuron. Indirect neurotransmitters are going to bind to G protein coupled receptors, or GPCRs. So indirect neurotransmitters are going to bind to G protein coupled receptors, or GPCRs. These GPCRs are then going to be activated, and they're going to elicit a signaling cascade that produces a secondary messenger. That secondary messenger is then going to bind to a channel-gated or a channel-linked receptor. It's going to open that channel, allowing ions to flow. So indirect neurotransmitters are going to bind to G protein coupled receptors, or GPCRs. GPCRs are then going to initiate a signaling cascade that produces a secondary messenger. That secondary messenger is what's actually going to go bind to that chemically gated ion channel, causing that channel to open, allowing for ions to flow in and out, and a graded potential to be produced. So GPCRs are going to be bound by indirect neurotransmitters. The process by which indirect neurotransmitters produce a graded potential is much slower than direct neurotransmitters because there's just more steps involved. So there are many, many steps involved in producing that secondary messenger, as opposed to in a direct neurotransmitter that just simply has to bind to those chemically gated ion channels. Indirect neurotransmitters are going to have a more prolonged effect on that postsynaptic neuron. The reason for that is because the secondary messengers are going to be prevalent for a longer period of time. So it's going to take longer for those secondary messengers to be removed from those chemically gated ion channels. So this is the signaling cascade, or an example of a signaling cascade that might be initiated when an indirect neurotransmitter binds to a GPCR. So we're going to walk through some of the key steps in this process. So an indirect neurotransmitter is going to bind to a G protein coupled receptor on that postsynaptic neuron membrane. So the indirect neurotransmitter binds to the GPCR on the postsynaptic neuron membrane. When that neurotransmitter binds to the GPCR, it activates the GPCR. That activated G protein coupled receptor is then going to activate a G protein. The activated G protein is then going to move across, and it's going to bind to and activate an enzyme in the membrane called adenylate cyclase. Adenylate cyclase, when activated, it's going to catalyze the conversion of ATP into a small molecule called cyclic AMP, or CAMP. Cyclic AMP, or CAMP, is then going to be that secondary messenger. CAMP is then going to go and it's going to change the permeability of the membrane to certain ions. So in other words, CAMP is going to come over here, it's going to bind to this ion channel, causing that ion channel to open. This then allows for ions to flow into or out of the cell, producing that graded potential. The graded potential could be excitatory or it could be inhibitory. So just like direct neurotransmitters could be excitatory or inhibitory, indirect neurotransmitters can also be excitatory or inhibitory. So I'm going to walk through this process one more time. So when an indirect neurotransmitter binds to a GPCR on the postsynaptic membrane, it activates the GPCR. Activated GPCR activates its coupled G protein. When this G protein is activated, the G protein can go across, bind to an enzyme in the membrane called adenylate cyclase. Adenylate cyclase is going to catalyze the conversion of ATP into a small molecule called cyclic AMP, or CAMP. CAMP is going to be that secondary messenger. CAMP can now come over here and bind to an ion channel in the membrane, causing that ion channel to open, allowing ions to flow into or out of the cell, therefore causing a graded potential. That graded potential could be excitatory or inhibitory. So we went over a number of different examples of indirect and direct neurotransmitters. A couple of examples of those indirect neurotransmitters are going to be some of our biogenic amines, so some of those feel-good neurotransmitters, some of those peptides, so our endorphins and enkephalins and cholecystokinin, and then some of those dissolved gases, so nitric oxide and carbon monoxide. In addition to neurotransmitters present at the synapse, we also have what are called neuromodulators. So neuromodulators, when released in the brain, are going to act on an area rather than just a single synapse. So they're going to affect the cellular properties or the synaptic properties at many synapses. So neuromodulators are going to alter the cellular properties or the synaptic properties. What that means is they might allow for that presynaptic neuron to release more neurotransmitter. They might cause that postsynaptic neuron to become more sensitive to that neurotransmitter. They may change the resting membrane potential of a group of neurons to make them more or less likely to fire. They might remove some neurotransmitter from the synaptic cleft in order to reduce the amount of neurotransmitter, and so on. So there's a variety of different effects of neuromodulators, but in general, they're acting on an area in order to alter the synaptic properties at many synapses or many neural pathways. We try to mimic neuromodulators in a clinical setting. So certain drugs are actually mimics of neuromodulators or mimics of these molecules that are changing the synaptic properties in the brain. So for example, two mimics of neuromodulators are selective serotonin reuptake inhibitors, or SSRIs. Serotonin is one of those feel-good neurotransmitters in our central nervous system. So serotonin is going to help you have this kind of high or this sense of feeling good. And so we actually use selective serotonin reuptake inhibitors in order to prevent the removal of serotonin from the synaptic cleft to treat depression. So we're trying to keep that high or that feel-good sensation by keeping serotonin present in that synaptic cleft. We also have cholinesterase inhibitors. So I've previously mentioned this. So in Alzheimer's, there's decreased levels of acetylcholine in certain regions of the brain. And so we have cholinesterase inhibitors that are going to come in and prevent the breakdown of acetylcholine in the synaptic cleft in order to keep that neurotransmitter around and therefore increase the levels of acetylcholine in order to counteract some of the negative effects of Alzheimer's, such as dementia. So cholinesterase inhibitors and selective serotonin reuptake inhibitors are just two examples of mimics of neuromodulators. Again, neuromodulators are acting on a large area in the brain, so they're trying to alter the synaptic properties of many synapses. So in both of these examples, it's altering the synaptic properties by preventing the removal of a neurotransmitter from the synaptic clefts.